So we were doing the experiments in tanks, in closed tank systems. So there was no chance of it getting back into the wild. We have to box up, container all the water we're using anyway, and we put it through a filtration system, and then finally put it through the sand as well, which acts as a natural filtration. So the antibiotics aren't going to get into the reef system as a whole, and it's all self-contained. So that's something we were very careful not to do, because we didn't want to produce these sort of superbugs, which might have even started occurring. Bacteria can particularly replicate and evolve really quickly. So even in that 6-day experiment period, we could have been evolving some different bacteria associated with those corals themselves. We actually developed quite an interesting thing. In Fiji, we adapted this a little bit more, and we used, I don't know if you get them here, Compeed blister plasters, like little gel plasters, and you can put the antibiotics on that, and you can actually put those around the coral, and it will start to emit the specific dose rate to the coral, so you don't actually need to continually dose. But again, we don't recommend we're going to do that in the wild, but it's just showing that it's got the potential to do that. And hopefully we can develop this, maybe a probiotic approach to it, but we will assess that as well quite carefully before releasing that into the field. You're going on the pessimistic side. No, that's true. So something I didn't mention was ocean acidification, another big problem facing reefs in general. And there are actually cases, there's like hydrothermal vents, which are very acidic, and you can actually have like conch shells where they're surviving, but they're really soft to touch. So ocean acidification is the next kind of buzzword and the big thing which people are starting to look at. The simple answer is we just really don't know. We have to hypothesize a lot of different things, but even with ocean acidification occurring, we're still seeing healthy reefs forming and young corals starting to recruit. They are under a lot of a variety of different stresses, as we showed there and as I showed last time. But they do seem to be quite tolerant. They seem to be able to adapt. How it's exactly going to pan out, I can only look into a mystic ball, but I think they're going to find a way. Like I say, it's going to change specifically, so we're going to get this variation. But I've never actually seen corals starting to dissolve on any rates which we've looked at as far as ocean acidification is concerned. If you scale it up right to the end predictions, in what we predicted in 2100 or something, then in tank experiments it's starting to occur quite a lot, and sometimes you see corals actually detaching. The polyps itself will detach from the skeleton and then land, and they can survive as a polyp, not as a colonial structure, for a little period of time until environmental conditions reduce back again. So maybe that might be a new evolutionary step which these corals are actually trying to see if that works. But they are suffering a lot of different stress events, and they are under a lot of pressure, but they seem to be really tolerant. I've just come back from Thailand doing some research out there, and the corals are surviving in a thick soup of water. There's areas where they have tidal exposure areas where the coral's actually exposed completely for lengthy periods of time, for like 5 hours, and obviously with that comes intensive solar radiation, immense heat, more so than what we get in the sea surface. Temperature rises. They cope with a little bit of solar bleaching on the west side, so they get a bit of bleaching, but the corals survive perfectly happy outside of water, which is pretty amazing adaptation to do. And there's certain species, one called the tombstone coral, which can actually retract its tissues down its skeleton and survive as a bare skeleton for a period of the time. And then when the tide comes in, it regrows over its skeleton in the space of a few hours and starts feeding again as normal. So there's some unique adaptations out there, and I believe the corals are going to find a way. We're going to lose some species for sure, but whether we lose all species or how that actually works, I can't say. But I think we're going to shift the ecosystem, but it will still be there, certainly in our lifetime, and hopefully for many other people's lifetimes in the future. But if we get a major ecosystem collapse, it's going to be a problem. And people are obviously, as I said right at the beginning, there's a lot of people who depend on it. Major food source for many, many countries, and again, a good source for gross national product and bringing in tourism and things like that. So hopefully we don't have to face that. But if we do, it's the same with rainforests, if we chop all those down. But I believe that something like, to put a dampener on things even more, I think something like a major human disease or food shortage or something like that is going to wipe out us as an entire race before we need to worry about complete ecosystems collapsing. Time or over the whole time. So there's a wide range depending on the actual disease we're looking at. Some diseases are quite persistent and can be found in low numbers, but throughout the whole year. Something like black band disease, you find maybe 2 or 3 on a reef tract, but they'll be around for quite a long period of time, and sometimes they can kill off that coral quite quickly. Other times they can go really slowly throughout the year and eventually spread on to other corals. Things like the dark spot in the Caribbean we saw, again, that's prevalent all year round. It doesn't seem to be as detrimental to the coral. The coral seems to just survive with these darkened patches for quite lengthy periods of time, and then all of a sudden they might lose a bit of tissue and then they carry on surviving again. Things like the white syndromes and the white bands, we do see a more increase in those during the warmer summer periods. So in the Maldives particularly, from about November, December till about now is the time where you see quite a significant increase in the disease. But you still can pick it up pretty much all year round. In the Maldives particularly because you've got less of a huge change with only this 2-degree kind of variation. You've got quite a high sea surface temperature anyway, so that will hopefully be providing these corals with quite a lot of resistance to increases in temperature, but it's also showing they're really on the top of their threshold, and any slight variation, like a particularly sunny day in the cooler months, then you'll still cause an additional stress and can cause the onset of these diseases. So yeah, there's the general pattern that you'll see more during warmer periods, but it's not necessarily a cut and dry case. Sorry, what? Fisheries. So that's something I've just come down from Corellian Lab this time, and I've had some students looking into it. And something I'm quite interested in is looking into things like parrotfish damage onto reefs. And it's just a hypothesis I'm working on at the moment, but the parrotfish seem to be causing quite a lot of fish bites. It naturally occurs anyway, but in this time round I saw a huge increase in the amount of fish bites, and that might be related down to the fisheries associated about local reef systems. And it might be a kind of catch-22 if particular fish species are banned in marine protected areas, you might get an increase. But also if you remove them, you also get problems as well. So if you remove them, you get a lot of algal growth. So it's like finding that balance, which is a very tricky thing to do. But I believe quite a lot of researchers are trying to look into where this balance is, and obviously create sustainable fishing catches and marine protected zones and things like that to accommodate everybody's needs, but also get to this balanced ecosystem where everything works how it should be. So we haven't really looked too much into the cross between fisheries and disease, but there are a few studies which have shown that certain species can actually transmit pathogens from one coral to another, particularly things like butterfly fish might be doing something like that. But it's probably not a big driving factor. The only thing is you're going to, if you fished out an entire group of, say, herbivores, for example, then you're going to have more competition from algae or natural predators like the crown of thorn starfish. That's thought to be caused by removal of the Titan triggerfish and also the Titan shell. If you fish those two main predators out, it's thought to be an example of why you might get an increase in the crown of thorn starfish and therefore cause an indirect effect of increasing coral death via that route. So there are slight linkages, but it's a relatively unexplored area at the moment, but something to look into in the future. Yeah, certainly it's an interesting question. So that was whether fish bites make corals more susceptible to coral diseases in general. And it's something I'm very keen to look into. What happens with the diseases, as I've hopefully shown, is that any kind of slight nick or damage to a coral, like anchor damage or tourists standing on it, that sort of thing, increases the opportunity for potential pathogens to come in. But corals are very good at healing themselves, and that's when you see those pink lines or purple structures occurring on corals. That's the immune defense of corals kicking in and fighting off things like burrowing barnacles, sponges, fish bites themselves. The coral can naturally defend itself to a degree if the rest of the environment is how it should be. If you've got an increase in temperature when there's a lot of fish activity, then yes, I believe the combined effect will increase the amount of coral disease you're seeing on these areas. And because visually looking at it, I think a lot of the parrotfish are tackling these Parietes species, which are the more tolerant species. It's giving a whole new suite of different Parietes diseases, which we've started noticing and started to follow this time round up at Corallian Lab. And there's quite a lot of Parietes die-off. There's a new disease, or a relatively new disease, called Parietes white patch syndrome, which was first described in the Indian Ocean in 2012, and something I've just started seeing in the Maldives as well. And we're currently working on that to see if we can figure out what's going on. So there's quite a lot in the pipeline. I'm working on about 12 different projects, which is keeping me a bit busy, along with teaching my classes in the university and applying for funds for more PhDs and postdocs and people like you guys to come and hopefully work at the lab and things. So watch this space, and in the next 6 months to a year, hopefully we'll have some positions and things for you up-and-coming marine biologists. That's the plan anyway. Anybody else? Anya? Different microphone.
Yeah. Thank you for a very interesting speech. My question is, the microbiota that is associated with the corals, will they differ depending on which part of the world you take it?
Yeah. So during my PhD, which was a fair few years ago now, I actually developed a little apparatus called the snot sucker, which quite a few people like the name of, but I got chastised by the old professors in the whole field. But they let me keep the name because they thought it was cheeky enough to get in. And that allowed us to put this apparatus over a whole coral, and we could compartmentalize the coral. So we could look at this sloppy mucus layer, this dynamic mucus layer I showed with the video. And then there's a thin gelatinous mucus layer as well, which is more sticky and thick and seems to be bacterial free. Then you get the tissue structure itself. Then you obviously get the Cymbididium as well, the algae. Then you get the skeleton, and then you get the gut mucus. And we could separate all those different compartments using this snot sucker. And we found that each compartment held a completely varied and distinct, significantly different microbial community on a healthy coral. We haven't replicated that onto diseased corals as well, but it's, again, something to take up and have a look at. But It shows it's a very complex environment, and this holobiont just gets even more confusing. And certainly when you start looking at fungi, which again is not really done, and then viruses is starting to become amazing, and it just carries on. Every time we answer one question, another million comes up.
Yeah, just one last question.
Yep.
Since you're associated with the Corallian Lab, I was wondering if you have facilities to identify microbes to the species level?
Not at the lab itself, but what we do is we take our samples, we take them back to the UK and analyse them there. So at the university we have in the UK, which has obviously got associations with the Maldives now, we can do a lot of microbial communities, so analysis down to species level, so a lot of sequence data. So if people are interested in getting involved in that, then give me a shout. I'll give some cards out if anybody wants to do that, and we can start looking into the practicalities behind that. Obviously, going down to the species level, the problem is funding. It gets quite expensive to do a lot of molecular work, but there is quite a bit of funding out there at the moment, and certainly a lot of student projects working together. If we start collaborations between students here and students at UK universities, we can find more funding to do projects such as that. So there's definitely possibilities, and we should have a chat about it. Anybody else? Well, thank you very much for coming. Hope you enjoyed it.